My name is Brianna Rivera, and the lab I work in is interested in a DNA repair protein called DNA Photolyase. The protein repairs a specific kind of damage caused by UV light on DNA using light-driven electron transfer to do so. We have a CPD lesion. The protein transfers an electron to it, and the DNA can repair itself, leaving you with repaired DNA. The entire process is driven by light, and the necessary cofactor, as found in the crystal structure here, is FAD. The cavity in the crystal structure is where the damaged DNA would bind, and the electron transfers between the colored species, which is the FAD cofactor, and the damaged DNA molecule occur. The colored species in the FAD cofactor, as seen in Figure 3, the specific states that we are interested in are the one-electron oxidized state, which is called the semiquinone state, and the fully reduced state. When we are heating the thermophilic version of the protein, we realized that something was happening in the pocket around the FAD cofactor. We can tell from the shift in the absorption spectrum when heating. The red peak, right here, sharpens and becomes more intense, and we are only looking at the semiquinone spectrum. There could be a few reasons for this change. One of the reasons I was examining was if heating the protein caused proteolysis of the protein. Proteolysis means that the protein was possibly cleaved into smaller pieces. So, we ran SDS page to monitor the size of the protein. What I have shown here in Figure 8 are 9 different protein samples, and in lane number 6 is the standard. On the gel, smaller proteins run faster and get further down the gel, while larger proteins run slower. We heated identical samples at 5 different amounts of time. The first sample in lane 1, right here, was the supernatant and was not heated. The second and third samples were the supernatant and the precipitate, respectively, and were heated for 10 minutes. Each successive sample is the supernatant and precipitated protein after 20, 30, and 40 minutes. This gel shows a very deep color band around 48 kilodaltons, which is our protein. As you can see, the protein doesn't change how it runs, so there doesn't seem to be any degradation of our protein in these samples, thus disregarding the idea that proteolysis is going on.
Thank you for taking time to listen to my presentation. Ariana Aguirre is going to talk about how it looks like the protein does seem to stabilize with heating.